also popularly elected speaker, is Jingjing Zhao, who will be speaking about how dyslexia genes influence brain structure and connectivity.
Oh dear. Hello everyone, I'm Jingjing Zhao from the Department of Psychology at the Chinese University of Hong Kong. Doesn't work? Yeah. I'll do it here. I have no disclosures, but I would like to thank all my co-authors. This project is collaborated with the University of Edinburgh during my sabbatical year there last year. For those who don't know what is dyslexia, let me give you an overview of what is dyslexia. Dyslexia is a heritable neurodevelopmental disorder that is known as a neural disconnection syndrome. And phonological deficit is taken as the key cognitive deficit of developmental dyslexia. And dyslexia is known as stem from difficulties in assessing phonological representations due to disrupted structural and functional connectivity among regions involved in phonological processing. Yet, a series-driven mapping of genetic risk onto brain structure and connectivity of neural hubs with associations to phonological deficit and reading phenotype of dyslexia remain a challenge. So my talk will cover all of this. And here is the framework that I start during the past decade. So we start from the cognitive deficit, and then the brain dysfunction, and then the genetic differences between control and dyslexia. And also I tap about some of the environmental factors. But in this talk, we only focus on these three factors. This is a paper published about 10 years ago, and they investigate the alphabetic languages. What is the cognitive deficit of developmental dyslexia from alphabetic language? They studied 6 languages in alphabetic languages and find out that actually there are 3 dimensions that is crucial for the cognitive deficit of developmental dyslexia, and include the rapid optimized naming, phonological awareness, and phonological memory. And this is the data from our lab. We published it last year. We basically find very similar results as alphabetic languages. It means that phonological deficit is also the key cognitive deficit in Chinese. And this is the brain level deficit that we discovered during the past decade. We focus on the brain connectivity deficit of developmental dyslexia. We think that developmental dyslexia is a disconnection syndrome. So the wet matter connectivity has some problems in developmental dyslexia children. And this is our finding. We find that the phonological area, that is the temporal lobe and the temporal occipital parietal subnetwork, is crucial for developmental dyslexia. And this is another hub-based wet matter connectivity network analysis. And we use this hub-based network connectivity, the hub nodal degree value, to calculate the nodal degree of each region of interest of the brain and see that whether it correlates with phonological ability of dyslexic children. And actually find out that in dyslexic children, the nodal degree of the left middle temporal gyres can predict their phonological ability. And then 2 years ago, I collaborated with the University of Edinburgh. We published a paper in Nature Genetics, and we observed that there are 42 genome-wide significant loci that is associated with dyslexia, and 15 of them linked to cognitive ability and educational attainment, and 27 are newly discovered genes. From there, we start to think about the next question, because now we find out that phonological deficit is the key cognitive deficit of developmental dyslexia, and it is a disconnection syndrome. And also we've observed the 42 genetic differences of developmental dyslexia compared to controls. So the next question is how dyslexic genes influence brain structure and connectivity, which can it be linked to phonological deficit and then to the reading phenotype. So in this study, we tested the associations between dyslexia-related polygenetic scores that we discovered 2 years ago and the gray matter structure and white matter connectivity of 85 brain regions of interest. And then we examined the brain structure and connectivity serve as intermediate endophenotype, mediating the effect of dyslexia-related polygenetic scores on dyslexia-related phonological phenotype and reading behavior. And we used 2 samples. One is the UK Biobank sample, included about 40,000 subjects. And the other one is the replication sample, which included the Generation Scotland sample, included about 1,000 healthy adults. And here is the pipeline of our study. We actually compute the polygenetic scores of all these subjects, and including not only their dyslexia-related polygenetic scores, but also dyslexia-related reading phenotypes and their phonological polygenetic scores. We calculated their brain volume, surface area, and also their cortical thickness of those 85 brain regions. And we also measured the nodal degree value of the connectivity of those 85 regions. And our idea is to see whether those polygenic scores correlated with their brain structure and whether these correlations can be validated on their white matter measures. And then if we find something interesting of the brain regions, whether this brain region can serve as the intermediate endophenotype to link with their phonological and reading phenotypes. Here is the result. So this figure is actually something like the Manhattan plot. If you study genetic studies, you will see Manhattan plot. And the x-axis in genetic analysis is actually the chromosomes. But here, in this plot, the x-axis is the brain regions, those 85 brain regions. And the y-axis is the p-value, that's the threshold. And there are two lines here. The blue line is actually the nominal significance level, and the red line is the Bonferroni corrected level. So we can see that there are many regions that surviving multiple corrections. And the letters are so small, but I can tell you that there are a lot of regions that is associated with reading and phonological processing areas, including the left fusiform gyres, the left superior temporal gyres, and also the left hexal gyres. So it means that we'll find something that's really interesting that is associated with polygenetic scores of dyslexia. And here is the different thresholds of the polygenetic scores of dyslexia. And the most important thing that I want to tell you is that we only observe that the polygenetic scores of dyslexia associate with brain volume, brain structure, and white matter connectivity, but not the other phenotypes, polygenetic scores of other reading and phonological phenotypes. So it means that most sensitive polygenic scores is dyslexia. And the most important, another important thing that I want to point out is that we find a similar kind of pattern from surface area, the correlation between polygenic scores of dyslexia associated with surface area of our subjects, and also similar pattern from cortical thickness. But across those three gray matter measures, the only one brain region that can be replicated across all those gray matters is actually the left insula, which is here. This is anatomy of the left insula. If you tease apart the left frontal gyres, you will see that the insula is right inside it. And it's a very important anatomical region that is associated with speech production, speech perception. And it includes two parts, the anterior part and posterior part. So left insula is an important region that we find out polygenic scores of dyslexia associated with its volume, its cortical thickness, and also its surface area. How about white matter? So this is a surface-based analysis of the left insula in our dataset. You can see that it's very significant, and it's most driven by the anterior part. And here is where the insula, left insula connected with all the other brain regions. You can see that it's connected with all those phonological regions and also those reading-related regions. And whether polygenetic score of dyslexia associate with white matter connectivity of the left insula. You can see that here is the UK Biobank white matter results, and all those measures of white matter, including their fractional anisotropy, streamline numbers, and also their mean anisotropy, and the others from the advanced DTI models, they are all influenced by polygenic scores of dyslexia. So this is actually the UK Biobank gray matter results, volume, surface area, and cortical thickness, and this is the white measure results. How about our replication sample? So this is our results of the discovery sample. And from the Generation Scotland dataset, we actually also replicate the results of the left insula. We find out that the polygenic scores of dyslexia associated with the volume of the Generation Scotland dataset, and also the surface area, and also streamlines numbers of this sample. So from here, we can see that actually polygenetic scores of dyslexia not only influence gray matter, but also white matter, not only from our discovery sample, but also from our replication sample. So how about the mediation? Whether the left insula, the gray matter structure, and white matter connectivity can serve as the intermediate phenotype that link between the polygenetic scores of dyslexia and phonological deficit, and also the reading phenotype? And the answer is yes. This is from the discovery sample. We can see that from the UK Biobank, the surface area, volume, and cortical thickness all can serve as an intermediate phenotype, and also the white matter connectivity measures of FA and SC. And in the replication sample, the Generation Scotland dataset, we have the reading phenotype, where the reading as the phenotypes. And you can see that it's a full mediation model. The volume, area, and streamline of the white matter measures can be an intermediate phenotype. So I think this is what I want to tell about the story. And actually, we observed that the several phonological and reading-related brain regions were identified, and only the left insula consistently show significant associations between dyslexia polygenic scores across multiple structure measures in both samples. And we think that this is very important to give an evidence for phonological deficit theory, because we have the direct evidence to see that how genetic influence the disconnection of the left insula, and then to phonological deficit, and then to reading ability. So that's about my talk. I think I don't have time, so there is some limitations, because this is an adult healthy sample, not dyslexia sample, and this is adults, not children. For most dyslexia, they are developmental children, so we want to see other evidence from developmental cohorts. And I think this is my talk.
Unfortunately, no time for questions because of our time constraint.
Okay.
But people wanting to talk to you can
yes,
talk to you
and I have a poster after this session, so you can come to my poster to have a more interactive discussion. So my poster number is 685. I'll see you there.